Okay, so I'm gonna go 3 pages at a time. So I'm gonna make 2 videos, so I don't rush through this with you guys. Make sure you watch this. Make sure you ask questions if you don't understand anything. All right. So we're gonna begin with the first page. What do we call the study of heredity? Well, that's genetics, how things are inherited. Okay. This diagram's kind of funky, and you'll see why with a D, but this is the only part that I really care about. For A, remember you've got two different stages of meiosis. You've got meiosis 1 and meiosis 2. In meiosis 1, what's happening, if you notice, you've got homologous chromosomes here. So as soon as you see two X's, those are homologous chromosomes. So in stage 1 of meiosis, the homologous chromosomes separate. And then down here in stage 2, if you see these are sister chromatid that make up the X, those are then separated in stage 2 of meiosis. So for A, this would be prophase 1. I'm not gonna get into super crazy details like I did in chapter 11, just knowing in general what's happening. So you see the chromosomes have formed, your spindle fibers have formed. Okay. B, they've lined up, so that's metaphase 1, okay, because we're about to separate the homologous chromosomes. Okay. In C, they separate, so that would be anaphase. Now I put anaphase / telophase because D, this is where it gets kind of funky. D is telophase, technically interphase, and then also prophase 2. So obviously on a quiz or test, I wouldn't ask you about D. I could totally ask you about this because you see homologous chromosomes lining up, so you know that it is metaphase 1. Okay. For E, you would know it would be metaphase 2 because you see the sister chromatid. So you just see one single X versus 2. 1, 2. Okay. So this is metaphase 2 because you have your sister chromatid lining up. Then when your sister chromatids separate, that's anaphase 2, and then you've got telophase. So as you notice here, the whole point of meiosis, and to keep meiosis and mitosis separate in your heads, in mitosis, you're just creating an identical copy. So you take a diploid cell, you make a copy of it, you've now got 2 identical diploid cells. In meiosis, you are taking a diploid cell, which you can see here because you've got 1 copy, 2 copy, you've got homologous chromosomes, and you're producing 4 haploid cells, and they are in no way identical. Now, these haploid cells make up our gametes, so they are either sperm or eggs, depending on gender. Okay, and that's it. Okay, really not that bad. Let's keep going. For here, it says decide whether the following are diploid or haploid. So remember, with diploid, that means you have homologous chromosomes. To have homologous chromosomes, that doesn't mean that you necessarily have the full X. This full X means that the chromosome has been copied or duplicated. So here, if you notice, I've got 2 pairs of a chromosome. They haven't been duplicated, so they're not duplicated, but one's from mom, one's from dad, therefore they are diploid. For here, I only have 1 copy of it because I don't have a homologous chromosome here. Therefore, it is haploid, but it is duplicated because you see that X. The sister chromatid represent duplicated chromosomes. Okay. Here, I've got 1, 2, 3. They don't have a pair. They don't make an X. Okay. So not only are they haploid because they don't have that other copy on the other side like they do here, but they also haven't been duplicated. Here, not only have they been, are there 2 of them homologous chromosomes, but since they make X's, they are also duplicated. Okay. For number 3, I messed up this question, so period 7 knows this, period 5 knows this, but period 7 doesn't. The question should say, how many autosomes do humans have? And now we're talking about a diploid cell, not a haploid cell. So we have 23 chromosomes. Okay, but 46 total because we have 23 pairs of chromosome, which make up 46 chromosomes if we have a diploid cell. So of those 46 total chromosomes, 2 of them are sex chromosomes, right? So either XX or XY. Okay? If you have more than that, you're an exception, you have a disease, and we don't assume that that's the norm. So we've got 2 sex chromosomes. So if 2 of them are sex, that means the rest, 44, are autosomes, meaning having absolutely nothing to do with sex, just regular chromosomes. Okay? This man is known as the father of genetics. That's Mendel. Okay, Gregor Mendel. He worked with the pea plants, just to remind you, possibly. Different forms of the same gene are called alleles. Okay. Here, this should be pretty nice and easy. Big A, since they're the same, they're homozygous, and it's dominant. This is heterozygous, and this is homozygous recessive. In gametogenesis, or the process of producing gametes, guys, are just eggs and sperm. Okay, this is also known as meiosis. So remember when I talked up here about the difference between meiosis and mitosis. Meiosis, we're making sperm and eggs. That's it. Okay, haploid sperm, haploid egg. In mitosis, we're making identical copies. Okay, if two alleles blend together, so red and white make pink, this process is called incomplete dominance. So it's not that you see the red and the white separately, you see them mixed together. That's incomplete dominance. If you see them simultaneously, like a brown and white chicken, that's codominance. Like a co-captain, you see both of them at the same time, that would be codominance. This should be a review at this point. The chromosomes to the right are known as, so this one and this one, that's a sister chromatid. What's represented here, guys, is that this chromosome has been duplicated to make this one. And so that's the process of DNA replication, which we studied in chapter 12. Okay. If I see two together, so that which has been duplicated and that one that's been duplicated, those are homologous chromosomes. Okay. Genes that are inherited together on the same chromosome, so they're located on the same chromosome, they're said to be linked. Okay. The only way to separate them onto a different chromosome is by a process called crossing over. Now, in the dihybrid crosses, what we're assuming is that our genes are not linked, that they're completely separate, and therefore, for example, this is my genotype for the mother, right? She's little b because she has white fur, and little r because it's smooth. What that's saying is this allele can go with that allele, or this allele can go with that allele. So it's not like they're linked in any way, right? Like a linked gene, they stay together. So these aren't linked. So when I'm creating my dihybrid cross, first you need to create the genotypes of the mom and the dad, which I did. The dad's heterozygous, so I wrote that out. Now, for the mom, she could be little b, little r, which I wrote there. She could be little b, that little r, which I did there. Little b, little r, little b, little r. So I wrote all of those out. For the dad, he could be big B, big R, which I wrote. Big B, little b, big R, or little b, little r. And so I'm showing all the different combinations because each of these alleles is not necessarily it doesn't stay with one of these alleles, and so I have to show all the different options. And so that's why you have a dihybrid cross. You have 16 different options of what would be created. Make sure you keep the letters together, guys, so you don't get a headache. Here are the genotypes. Remember, genotypes are just showing the alleles. It doesn't say what it's going to look like. It's just showing the different alleles. So here are my probabilities. Let me know if you disagree with anything, and you can pause and go back and look at my dihybrid cross. For phenotype, it's what they actually look like. So for example, this one would be black and rough. So a quarter of them are going to be black and rough. Here, that's going to be black and smooth. So that's another quarter. Okay? It's not too bad, hopefully. All right. Same. We're going to do another dihybrid cross. I'm not going to walk you through it quite as much. This is the genotype of the mom. This is the genotype of the father. And so I need to see all the different combinations. So this little n could be with this big B, little n with little b, little n with big B, little n with little b. And so I showed all of those right there. Okay. Now for dad, big N with little n, big N with little n with little b, little n with little b. And so I showed all those options there. And then I combined them together. So same thing as up above. Here are my probabilities. You can check them out. Pause me. Okay. All right. So the process that's shown here, so we've got two homologous chromosomes. They hold hands, okay, which, by the way, is called synapses. We'll get to that in a second. But once they've held hands, what can happen is that the genes here and the genes here can flip. Okay, so this, you can't see it unfortunately, but this has new genes, this has new genes. That process is called crossing over. That's the only way of getting linked genes to become unlinked. Okay. Now in the point of attachment, where the spindle fiber, for the spindle fibers, right at the center of sister chromatid, right here, that's called the centromere. I just did 10 with you. It's called synapses when they're holding hands. Okay. And then 11, this last one, this principle states that alleles separate when gametes are formed. Okay, this enables us to create a Punnett square. So just the fact that, okay, the mom is big A, little a, but those don't stay together. Instead, they're going to separate out, and a kid can either get that big A or that little a. From the dad, the little a and the little a, the kid's not going to get both from the dad, just one. So they separate out. That separation of the alleles, that's law of segregation. Okay, and then we'll get to law of independent assortment in the next video.